So, good afternoon, guys. I am Rishabh, and today we'll be starting with the week one of the course, that is host pathogen interaction by Professor Himanshu Kumar from ISER Bhopal, and I'll be one of the PMRFTAs from the group, and we'll discuss a couple of questions related to the week one in this lecture. And I prefer, if you guys have any doubt, you can ask me in between itself. Okay, so let's start. So the first question for today is, which experimental approach proved that serum from animals exposed to toxins could protect other animals? So what can be the correct answer? Okay, so the correct answer is option 2, that is serum therapy experiments by Behring. Okay, so here is the explanation for that. So Behring actually used guinea pigs. Okay, so he used guinea pigs and he injected a, a serum from the affected diphtheria guinea pigs, and suggested that the pigs that received the serum from the affected guinea pigs developed immunity against the diphtheria toxin. Okay, so it was the first example for the prophylactic serum therapy, and he also received a Nobel Prize for his diphtheria anti-serum discovery, and he termed the serum as antitoxin. Okay, so here was the experiment that he conducted. So in one of the experiments, in experiment A, he took uh, serum from an uninfected guinea pig and exposed in the other pig, another normal pig, the same serum. Okay, and then when the pig was affected by diphtheria, it died. Okay, so why it died? Because it doesn't have any immunity against the diphtheria toxin. In other experiment B, when uh, a guinea pig was initially induced with diphtheria, but but then the serum from this particular strain was injected into the other normal guinea pig, and after the diphtheria was induced, the guinea pig was normal. That means that that means that uh, the pig actually developed immunity against the diphtheria toxin, because it was initially had been transferred from a strain that was already infected with diphtheria. In the other experiment, the guinea pig was a guinea pig was induced with another bacterial strain that doesn't cause diphtheria, and when its serum was transferred to another guinea pig, then when it was injected with a lethal dose of bacterial strain A that causes diphtheria, then the guinea pig died. Okay, so why it died? Because it doesn't have immunity against the diphtheria toxin bacteria. Okay, so these suggest that whenever we have a disease, or the pigs have a disease, if we actually take out the serum from those particular pigs and inject into a normal pig, so this pig would have immunity against the diphtheria toxin. Okay, so that's why these were called as antitoxins, and here is the experiment for how it performed. Okay, so if you guys have any doubt, you can ask. Okay, if no doubt, then we'll move on to the next question. What is the primary purpose of the Gruber-Vidal test? What can be the correct answer? Okay, so let's see the options. It is used to detect antibodies against Salmonella typhi in the patient's serum, to diagnose typhoid fever, to identify bacterial toxins in the blood for rapid typhoid diagnosis, to observe to observe bacterial growth patterns in blood samples, to differentiate between typhoid and other bacterial infections using heat stable antibodies, and to detect complement activation leading to bacterial lysis in typhoid patients. Okay, so the correct answer is option A. That means to detect antibodies against Salmonella typhi, that is the major causative organism of typhoid, in the patient serum to diagnose typhoid fever. Okay, so Gruber-Vidal test is basically used to detect whether the patient is suffering from typhoid or not. Okay, so typhoid is caused by a bacteria called as Salmonella bacteria. It has two strains, Salmonella typhi and Salmonella paratyphi. Okay, Salmonella typhi expresses two antigens, antigen O and antigen TH. Okay, then we have the Salmonella paratyphi that expresses antigen AH and antigen BH. Okay, so now how does this basic experiment happens? So we have purified Salmonella typhi and Salmonella paratyphi antigens. Okay, that we keep on a plate, and then on this plate we add the serum from the patients. Okay, so now when we add serum from these patients, so they actually, the if the antibody would be present in the serum, then it would bind with the antigen of the Salmonella typhi. Okay, so this antigen antibody reaction which will cause clumping. Okay, so this clumping will appear like this. Okay, so here as we can see, if so when this antibody will bind to the antigen, it will cause clumping. And here we have shown the reaction with the all the antigens that are expressed by the Salmonella typhi strain. So this is the antigen O of the Salmonella typhi, antigen H from the Salmonella typhi, and AH and BH from the Salmonella paratyphi. Okay, so this, as you can see here, these dots are called as clumps. Okay, so if the patient is positive with the typhoid, then the antibodies will be produced in the samples and that will bind to these antigens. Okay, so if these clumps are there, then O and H. For example, this O and H shows the clumps, so these are affected by Salmonella typhi. AH and BH do not show any clumps. Okay, so they are unaffected by the Salmonella typhi, but Salmonella paratyphi. Sorry. Okay, and this is the negative control. As you can see here, the negative control and the paratyphi strain ones are similar, and this is the positive control. As you can see, these dots as it appears in the O and H spot. So okay, so this is how we detect whether the patient is suffering from typhoid or not. Now, this was a qualitative essay. Now, with the advancement in science, we are able to quantitate this also. Okay, so quantitative assessment includes the serial dilution of the serum, okay, with the antigen. So when we can actually test the amount of keeping the amount of antigen constant in all the tubes, when we dilute the serum serially diluted, then it can actually tell us what is the titer of and what is the titer of the antibodies that are present in the patient sample. Based on that, we can detect the severeness of the disease. Okay, so this is how we can do the Gruber Vidal test that will be able to tell whether the patient is suffering from typhoid or not. Okay, so I hope this would be clear. Okay, so let's move on to the next question. Which of the following statements best reflects Buchner's 1889 discovery regarding alexin in blood serum? So, what can be the correct answer? Okay, so the correct answer is option 2. That means Buckner demonstrated that alexin could destroy bacteria independently of the immune cells. Okay, so what he did was he actually isolated a substance from the mice body. Okay, a substance that was heat labile. That means sensitive. So it was heat sensitive at 55 degrees Celsius, and this could destroy the bacteria independently of the immune cells. Okay, so this was the major finding from Buckner. So in 1889, that states that when there is a compound called as alexin that is present in the blood serum, and that is heat sensitive, and that can easily destroy the bacteria independently of the immune cells. And later this term was given by complement. So later it moved to the complement system, that it can directly act on the bacteria irrespective of any of the immune cells that are present in the body. Okay, so this was Major Buckner's statement from the view. So if you guys have any doubt, you can ask. Okay, so let's move on to the next question. What were the key observation in the Pfeiffer experiments? So what can be the correct answer? Okay. So the options are Vibrio undergoes phagocytosis in immune animals. Vibrio are killed by immune cells in the peritoneal cavity. Vibrio undergoes bacteriolysis in immune animals, swelling, dissolving, and disappearing within 20 minutes. Immune serum prevents Vibrio from multiplying in test tubes, and Vibrios are engulfed and destroyed by cellular activity in immune animals. So the correct answer is option 3. That means it undergoes bacteriolysis in immune animals, swelling, dissolving, and disappearing within 20 minutes. Okay, so as we can see here, so his work was majorly on Vibrio cholerae that basically caused cholera. Okay, at that time, cholera was the major disease that killed people. Okay, so he worked on cholera that was caused by Vibrio cholerae, and it was a comma-shaped bacteria, as you can see here. So these are comma-shaped bacteria that causes cholera. Okay, so his work was when mice were injected with Vibrio cholerae in peritoneal cavity. Okay, so what happened? Because they had the immunity, and this immunity, the serum or the immunity, actually helped to destroy these vibrio by swelling them, dissolving them, and disappearing them within 20 minutes after injection. Okay, so already immune mice that have the immunity against Vibrio cholerae, when Vibrio was injected in the peritoneal cavity, then these Vibrio disappeared within 20 minutes. Okay, and then he actually named this serum or the phenomena as endotoxin, so that this bacteria is actually disintegrated because of this endotoxin. Okay, so this was the experiment done by Piper. Okay, so if you guys have any doubt, you can ask. So let's move on to the next question. Which of the following concepts have contributed to the foundational theories of immunology? Select all that apply. So what can be the correct answer? So the correct answers are 1, 2, 4, 5, and 7. Okay, so Metnikov's phagocytic theory states that the cells basically from the sponges, okay, so they can actually engulf the different pathogenic organisms. Okay, then airless side chain theory. This leads to the this is another theory. So this theory basically states that there are certain receptors that can actually bind to the toxins on the surface of the cells. Okay, so this is basically the receptors that can actually bind to the toxins and that can lead to immune response. Okay, so then these receptors actually gave rise to the antibodies concept. Then the complement system. So this complement system is a system that is present in the body, that is present in the body that is connecting link between the innate and adaptive immune system. Then the Behring serum therapy. Behrings actually suggested that serum from a, as we have discussed in the previous slides also, that serum from affected animals, if given to the unaffected animals, then they can develop the immunity against the disease. Okay, then the dichotomy between the cellularist and humoralists. This is because some of the scientists considered that that cells are responsible for the immune system, but the other in by the Ehrlich cytine theory, it basically evolved that the antibodies also help in the immune response. So both of the scientists have their own terms about the immune system. So all these apply in the single immune system. Okay, so let's move on to the next question. What did Edward Jenner was used to develop the first successful vaccination? So what can be the correct answer? So the correct answer is option B, that is cowpox virus. Okay, so at that point of time, the smallpox was actually so the smallpox was actually very evident. So the scientist Edward Jenner was actually saw that in cows there was another similar type of disease that was following, that was cowpox. Okay, so he actually took out serum from the cows and injected in the patients who are suffering from smallpox, and they recovered. Okay, so the exposure of cowpox protected the individual against the more dangerous smallpox, and then this is how the first vaccination that took place was demonstrated by Edward Jenner. Okay, so let's move on to the next question. What is the main difference between the cellular and humoral theories of immunity? So, what can be the correct answer? So the options are: cellular theory emphasizes immune cells, while the humoral theory focused on soluble factors in the blood. Cellular theory highlighted antibodies, while humoral theory focused on phagocytosis. Okay. So the first option is correct, the second option is false. Then cellular theory concentrates on complement proteins while humoral theory does not. No, this is also false. Cellular theory is applied to bacterial diseases while humoral theory is applied only to viral infections. No. Cellular theory ignored the role of macrophages while humoral theory emphasized their importance. No. So basically cellular means that the cells that are involved in the immune system, and humoral theories which means that the soluble proteins or the antibodies present in the blood that are involved in the immune system. So the correct answer is option number one. Okay, so these were the major scientists that believed in both the cases, starting with the Metnikov. So these were the cellularists, and Ehrlich, that was humoral. Okay, so humoral immunity. So starting with Charles Darwin, they actually focused that phagocytosis general mechanism for the host defense. Moving on to the Louis Pasteur, both yeah, he believed in both the cases that the infection biology. Same with Robert Koch. Then Rudolf Virchow, these he suggested that cells are central to the immunity. Then Coleman, phagocytosis and inflammation, followed by starting from Emil Fischer, the key lock principle and differential chain as than the side chain theory. Then starting with the Behring, he suggested the antigen antibody reaction. Okay, so this is how some of the scientists believe that only the cells are a major part of the immune system, while the other believe that the antibodies are the major components of the immune system. Okay, so let's move on to the next question. Which type of immune specificity is primarily associated with innate immunity? So, what can be the correct answer? So the options are: narrow specificity for unique molecular structures, broad specificity for molecular patterns, memory specificity for prior infections, tolerance specificity to avoid self-reactivity, adaptive specificity that develops over time. So the correct answer is option number two, that is broad specificity for molecular patterns, because innate immunity is actually broad or non-specific. Okay, so it will depend upon only the molecular patterns that are present on the pathogen. Okay, so that will give us the immunity and its types. Which of the following contributes to the regulation of immune responses? So, what can be the correct answer? So the correct answers are 1, 3, and 4. Okay. So pro-inflammatory and anti-inflammatory cytokines. So basically regulation here means either increase or decrease in the immune response. Okay. So pro-inflammatory cytokines will increase the inflammation, and anti-inflammatory cytokines will actually decrease inflammation. Okay, then we have the third one, that is positive and negative feedback mechanisms. So in this case, The positive feedback mechanism will lead to increase in the amount of immune response, and the negative feedback mechanism will lead to inhibition of the immune response. Then cellular interactions as M1 M2 balance, M1 M2 macrophage balance. So M1 is a inflammatory cytokine. Okay, so this is sorry, M2 is an inflammatory cytokine and M1 is its regulator. Okay, so whenever there is there is some stimulus for the inflammatory site macrophages to be developed, M1 then is moved to M2, and if the inflammation gets over, then it is converted back to M1. Okay, so this is how the regulation in the immune response takes place. Okay, so let's move on to the next question. This work led to the development of the first vaccine. So what can be the correct answer? So it was Edward Jenner. Okay, so the correct answer is option 3, Edward Jenner. So he developed the vaccine for smallpox using cowpox virus. Okay, so let's move on to the next question. Which of the following statements is or are incorrect? So the correct answer is 2. That means first and second are incorrect. Okay, so let's see about the smallpox disease. So this is a virus that emerged during the causing illness and death in human population, and it is a double-stranded DNA genome that ranges from 130 to 230 kilobase pairs. Its symptoms include high fever, head, body aches, sometimes vomiting, and disfigurement and death. Okay, so this is the path course of the disease. So in by day 4 there are some screen spores that are filled with thick opaque fluid and liquid that have a dent in the center. Then moving to day 6, those rash become the sores become pustules, and after that, by the end of the second week, the rash appears, most of the sores are scubbed over. Okay, so this is the course of the smallpox disease. Okay, so let's see the options. Smallpox is caused by a double-stranded RNA virus. No, this is wrong. Okay, so it is double-stranded DNA virus. Smallpox claimed a lot of lives in Europe and was called as a White Death. No, White Death was called as plague. Okay, plague disease caused an epidemic in Greece during 430 BC. So yes, this is correct. Okay, so the first and second option are false or incorrect. So the correct option is second. Okay, so let's move on to the next question. Metnikov's, so which of the following statements is or are correct? So what can be the correct answer? Okay, so the correct answer is option number 5. That means all the three options are correct. Okay, so let's see the options. Metnikov hypothesized that cell are the major effector of immunity. Yes, so he actually proposed the phagocytosis theory. Okay, Koch postulated that the microorganism must be found in diseased but not healthy individuals. Okay, so yes, this is also correct. So Koch actually postulated some of the postulates that relates to the causative organism, and one of the postulates was that if a microorganism is causing the disease, that means this is actually present in the diseased organism, but not in the healthy organism. Okay, and it can be isolated from this particular diseased organism and cultured outside. So this was also one of the postulates from the coach. Then exposure to cowpox can protect individuals from smallpox. Yes, this is also correct. This is the method that Edward Jenner actually used to develop the first vaccination. Okay, so that's why all the three options are correct. Okay, so let's move on to the next question. Which of the following statements is or are correct? So, what can be the correct answer? So, the correct answer is option number six. That means only third is correct. Okay, so let's see the options. Hmm. So, the Gruber-Vidal test is used to diagnose diphtheria. No, it is used to diagnose typhoid. Okay, Alexander Yersin first isolated and characterized a toxin from Yersinia pestis. No, this was also not correct. So, Yersin didn't isolate and characterize a toxin from Yersinia pestis. The bactericidal power of defibrinated blood against anthrax bacilli was destroyed by heating to 55 degrees Celsius, according to George Natta. Yes, this is correct. So, what he did was he isolated the blood that was actually being suffering from anthrax, and when it heated at 55 degrees celsius, the power or the power to destroy this particular bacteria destroyed. So this was the feature that was described by the nutter. So the third option is correct. The correct answer is option number 6. Okay, so let's move on to the next question. Which of the following statements is or are correct? So, what can be the correct answer? So the correct answer is option number 8. That means none of the statements are incorrect. So let's see. The side chain theory of antibodies was proposed by Paul Ehrlich. Yes, this is correct. Then Emil von Behring termed the serum as antitoxin against diphtheria. Mhm. Yes, this is correct. Okay, then Klebs and Loeffler first isolated corynebacterium bacterium diphtheria, and that is diphtheria bacillus. Yes, this is also correct. So the correct answer is, that means all the three statements are correct. None of the statements are incorrect. So the correct answer is option 8, that is none of the above. Okay, so let's move on to the next question. Which of the following statements are correct? So what can be the correct answer? So the correct answer is option number 3. That is, first and second statement is correct. So let's see the statement. Edward Buckner termed the substance in the blood stream that has antibacterial activity as uh, elixin. Okay, so the first statement is Edward Buckner termed the substance in the bloodstream that has antibacterial activity as elixin. No, this is not correct. Hans Buckner, his brother, actually gave this thing that the antibacterial activity of the blood is elixin, and this is now termed as complement system. Okay. Edward Jenner first reported to the Royal Society of Oriental Practices of variolation of smallpox. No, this is false. Then, according to Koch, the microorganism must be cultured from the diseased individual. Yes, this is correct. Okay, so the correct answer is only third, not first. So, third only. Option 6 would be correct. Okay, sorry for the misprint. So, it would be option 6. Okay, so let's move on to the next question. Which of the following statements is or are incorrect? In an allogenic transplant, the donor is an identical twin. Comparative immunology deals with the immunological relationship between developing fetus and the mother immunity, and systems biology focus on the cells and the molecules, how they respond to vaccines and infections. So, what can be the correct answer? Okay, so the correct answer is option 2. That means first and second are incorrect. So, in an allogenic transplant, the donor is an identical twin. No. In an allogenic transplant, comma, the donor is actually a person who is not related. Okay. Then comparative immunology deals with the immunological relationship between developing fetus and mother immunity. No. This is not the case. Comparative immunology basically deals with the immune response between two species. Okay. Systems biology focuses on the cells and the molecules and how they respond to vaccines and infections. Systems biology focuses on the cells and the molecules and how they respond to vaccines and infection. So yes, this is correct. Okay, so the correct options are first and second, that they are incorrect. So correct option is option number 2. Okay, so let's move on to the next question. Which of the following statements is or are incorrect? So what can be the correct answer? So the second statement is incorrect. The persistence of antibody upon vaccinia virus is longer than the influenza virus. Yes. So what happens is in case of the vaccinia virus, the immunity or the antibodies can actually be present till many years, maybe around 10 years of age. Okay, and in influenza virus, it's in months. Okay, the post-transcriptional long regulation of non-coding RNA is an example of tight regulation of immunity at the cellular level. No. Okay, so at the cellular level, it's the cytokines basically, not the non-coding RNAs. Okay, so the second option is incorrect. So the correct answer is option number 3. Okay, so let's move on to the next question. Which of the following statements is or are correct? So what can be the correct answer? Okay, so the correct answer is option 8. That means none of the statements are correct. So let's see. Flagellum from gram positive bacteria sensed by our immune system in X. Follow. Narrow range diversity hallmark of the immune system? No. Okay. So the flagellin is basically is detected by the innate immune system. So it is an example of broad range. Okay, immune cells but non-immune cells express pattern recognition receptors. Okay, so only immune cells express PRRs. Okay, and the immune cells can express pathogen associated molecular patterns. No, PAMs are basically expressed by the pathogens or the microorganisms. Okay, so all the three statements are incorrect, so none of the statements are correct. So let's move on to the next question. Okay, so the correct answer is option number 7, that is second and third are incorrect. Okay, so let's see the explanation. MALT, that is mucosal associated lymphoid tissue, is an example of the peripheral lymphoid organ. Yes, this is a correct answer. Okay, the primary lymphoid organs include B, bone marrow, and thymus, while this one goes into the periphery lymphoid organs. Okay, the MALT. Then we have the bone marrow develops into a major lymphoid organ at 2 weeks gestation. No, this is also false. Okay. Then we have spleen is present in the upper chest location of the body. No, it is present in the that is near to the stomach or the pancreas. So this will be in the abdominal cavity. Okay. So that's why these two options, second and third, are incorrect. So seventh option is correct. Okay. So any doubts in this particular question? Hmm. Will it be closer to the lung? Wait. Spleen is not located in the chest cavity. That is the thoracic cavity. It is located in the upper quadrant of the abdominal cavity. Okay, so that is why it is not being it will not be able to it will not be present in the upper chest location. Okay. Okay, so let's move on to the next question. Which of the following statements are incorrect? So what can be the correct answer? Okay. Any more answers? Mhm. Okay. Any more answers? Okay. So the correct option is option number five. That is, all the three are incorrect. So let's see the statements. Bear's patches are predominantly located in the large intestine. No, they are present in the small intestine. Okay, so this is the location of the Peyer's patches. These are present in the small intestine. This is also a type of the peripheral lymphoid organ. Then the interaction of antigen to naive immune cells takes place in thymus. No. In thymus, differentiation of T cell takes place. So those are not naive. Okay, so that's why the thymus is a secondary lymphoid, is a primary lymphoid organs where the differentiation of the T cells actually happens. Okay, then fetal pancreas is the major lymphoid organ in the third month of the gestation. No, this is false. So all the three statements are incorrect. That is why option number five is correct. Okay, yeah, just 1 minute. Someone said something. No, actually, naive T cells are not introduced in the thymus. Okay, so they basically differentiate into different types of T cells. Okay, so that is not the work of the thymus. So that's the case. The naive T cells actually help are present in. The naive T cells actually encounter with the antigen in spleen and the lymph node. Okay, so only the thymus and bone marrow helps in differentiation. So that is the case. Okay, and in this figure we can see the position of the Peyer's patches also. So this Peyer's patches is present near the small intestine area. Okay, so and in not in the large intestine area. Okay, so this was the last question for today. If you guys have any doubt regarding any question, we can discuss or from any of the components of this week. Okay, if no doubts, then we'll close the session for today. Thank you guys for joining in. Let's meet next Sunday for the second week. Okay, thank you.